So we'll start with microbiology discussion on morphology and physiology of bacteria. So first we'll start, what are the differences between your prokaryotes and eukaryotes? Okay, so your bacteria comes under prokaryote. So we'll learn about the differences of prokaryote and eukaryote. Prokaryotes are unicellular. Eukaryote are multicellular. And they lack nuclear membrane. Prokaryotic organism doesn't have nuclear membrane, poorly defined due to absence of nuclear membrane. Eukaryote has well-defined nuclear membrane. Nucleolus is absent in prokaryote, which is present in eukaryote. Other organelles like mitochondria, Golgi body, these all are absent in prokaryote, which will be present in your eukaryote. Bacteria and blue-green algae comes under prokaryote. Fungi, plants, other animal cells comes under eukaryote. So question has been asked, which are the prokaryotes? So the answer was your bacteria and blue-green algae are prokaryote. So this was asked in PGI around 5-7 years back. This question was asked. The answer, PGI will get multiple correct answers. So the answer was bacteria and blue-green algae. Now coming to resolution of light microscope. Please remember, resolution of light microscope is 300 nanometer. We know the type of microscope. We have light microscope, we have phase contrast microscope, okay. Then we have dark ground microscope or dark field microscope. Then we have electron microscope and fluorescent microscope. Okay. So coming to resolution of light microscope, which is 300 nanometer. A phase dark ground microscope demonstrate the flagella. Flagella is a organ of locomotion of bacteria. To become motile, the bacteria has to have flagella. Okay. So dark ground microscope is used to demonstrate motility of bacteria or to visualize flagella of bacteria. So this year you had a neat question, right? Related to Treponema pallidum, where you visualize this bacteria. So the answer is your dark ground microscope. Then fluorescent microscope, you use fluorescent dye. Fluorescent dyes are auramine and rhodamine. Negative staining. Negative staining when your background has stained. Okay, your background has taken stain, but organism has not taken stain. So that is your negative staining. Okay. Example for negative staining is your India ink. India's ink, which we use for capsular staining. So that also has been asked previously, which negative staining or which staining which we do to demonstrate capsule of the bacteria. So India ink, your capsulated organism like your Streptococcus pneumoniae, Klebsiella pneumoniae, Cryptococcus, you have to demonstrate the capsule with the help of India ink. So which takes stain? The organism doesn't take stain, your background takes the stain. Now coming to what are the differences between the cell wall of your gram-positive and gram-negative bacteria, right? Because the property of gram-positive and gram-negative differs. That is because of their cell wall difference and few other differences apart from cell wall. So peptidoglycan layer, both have peptidoglycan layer, but gram-positive have much thicker peptidoglycan, 60 to 80 nanometer than gram-negative, which has 2 nanometer. Because of this thicker peptidoglycan, they take the gram stain and does not lose. Okay, so they take the crystal violet or methyl violet, gentian violet, so they appear violet in color. Okay, because of thickness of their cell wall. Now coming to lipid content. In gram-positive, it's almost negligible or nil lipid content. In gram-negative, you have lipid around 50 to 20%. Lipopolysaccharide will be absent in gram-positive, which we say LPSL. So we say lipopolysaccharide, which is present in gram-negative. The endotoxic property, endotoxin production in gram-negative is because of this lipopolysaccharide. Now teichoic acid present in gram positive, absent in gram negative. Aromatic amino acid, which is absent in gram positive, present in gram negative. Porins. Porins are absent in gram positive. Porins are present in gram negative. Okay, so this has been asked, what are the teichoic acid, where it's present, gram positive. Porine, where it's present, outer membrane of gram negative bacteria. So this is the picture of your gram positive bacteria, and this is the picture of your gram negative, which has lipopolysaccharide layer. Now, how the flagella? Flagella is the organ of locomotion of bacteria you need for the motility of the bacteria. So how many types of flagella is there? The bacteria who doesn't have flagella, we say it as a atrichous. So this is atrichous, okay, which has single flagella at one end. We say it as a monotrichous. So it has been asked, bacteria with single flagella at one end is called as various option. So correct answer was monotrichous. Where we see monotrichous flagella? So we see in Vibrio cholera, we see in Pseudomonas, we see in Campylobacter. So Vibrio cholera, Pseudomonas and Campylobacter has monotrichous flagella. Lophotrichous, where tuft of flagella is present at one end, which we see in Spirillum. Peritrichous, which we see in Salmonella typhi or Escherichia coli. This is the example for your, okay, peritrichous. Throughout the body, the flagella is present. So that is called peritrichous, which we see in Salmonella typhi or Escherichia coli. When flagella is present at both ends, we say it is an amphitrichous. Amphi means both. So both end flagella, amphitrichous, which we see in Alcaligens fecalis. Okay. So please remember, monotrichous, single flagella at one end, lophotrichous, tuft of flagella at one end, peritrichous, flagella present throughout the body, amphitrichous, tuft of flagella at both ends. Amphi means both. Okay. Now, which all are capsulated bacteria? That also has been asked previously. So just remember, pneumococcus, meningococcus, Haemophilus influenzae, Klebsiella pneumoniae, Pseudomonas aeruginosa, Bacteroides fragilis, Bacillus anthracis, Streptococcus pyogenes. So these can be capsulated bacteria. Okay. So most of these bacteria, their capsule is polysaccharide in nature. Few exception is Bacillus anthracis, where capsule is polypeptide in nature. In Streptococcus pyogenes, it is hyaluronic acid. Okay, so all has polysaccharide capsule, but you have polypeptide capsule in Bacillus anthracis. Please remember the name of capsulated bacteria, which has been asked previously. Which of them are capsulated except? Now coming to bacterial growth curve. The plenty of question has been asked previously on bacterial growth curve. So this is the bacterial growth curve, number of bacterial cell and time. So this is your lag phase. Lag phase, then you have log phase, then you have stationary phase, and you have decline phase. Okay. So lag phase is the phase where bacteria adapts to new environment. Okay. And they attain maximum size. Okay, adjustment to environment, all protein and other things, substance synthesis happens in lag phase. They prepare themselves, okay, and they become bigger in size, but number does not increase, size only increases. Then you have log phase, which is exponential phase. Bacteria divide and number increases. Then you have stationary phase. Stationary phase is the phase where you see spore formation. Sporulation happens. Things will happen in stationary phase. Inclusion body formation. These all will happen in stationary phase. Log phase is the phase where bacteria takes gram stain very well. Other phase also bacteria takes, but stationary and all, they won't be uniformly stained. In log phase, bacteria gets uniformly stained with your gram staining. Okay. Decline phase is the phase where you see involution forms. Okay, L forms and other are involution forms. That involuting forms you see in a phase of decline. So you have lag phase where bacteria prepare themselves to external environment. Log phase where they divide maximum. And stationary phase where spore formation or inclusion body formations Polymetaphosphate formation will happen in stationary phase. Decline phase, where involuting forms will form, L forms and other forms will form. So on the basis of temperature, how we divide the bacteria, what all terms we use. Psychrophile, which grow best in less than 20 degrees centigrade. That is, example is your Pseudomonas. Mesophile, which grows as 25 to 40 degrees centigrade. So most of your pathogenic bacteria comes to this group of mesophile, 25 to 40 degrees centigrade. Thermophiles, which takes higher temperature, 55 to 80 degrees centigrade. That is your Geobacillus stereothermophilus. Okay, psychrophile less than 20, mesophile 25 to 40, thermophile 55 to 80 degrees centigrade. Now coming to molecular method, what all molecular techniques which we use in microbiology, right? The molecular techniques are more in a picture after the HIV outbreak, COVID outbreak. The molecular method has gone very significance, very high significance. So molecular method, you have PCR, polymerase chain reaction. You have real-time PCR, you have loop-mediated isothermal amplification, which is LAM. Then you have biofire film array, which is called multiplex PCR. Then you have CBNAT, which is cartridge-based nucleic acid amplification. So in PCR, 3 steps is there. Okay, you denature the DNA first. So first will be your denaturation, which happens at higher temperature. That is, you do at 95 degrees centigrade temperature. Denaturation. Then you will have next step, annealing. Annealing happens at the temperature of 62 to 65 degrees centigrade. Okay. Annealing, where primer comes and start binding to the strand. Then extension, where DNA polymerase start extending the fragment of DNA, that is extension. That will happen around 55 degree centigrade. So you have PCR, denaturation, annealing, extension. Okay. Then what other microbial typing technique you have? You have phenotyping technique, you have genotyping technique. Please remember these all are probable question, because in microbiology, the molecular diagnostic technique has taken very high importance. So what are the phenotypic method? You have bacteriophage typing, which we do for Salmonella, Staphylococcus and Vibrio cholera. Bacteriocin typing, which we do for Shigella soni and Pseudomonas. Biotyping is done for Corynebacterium diphtheriae and Vibrio cholera, which you have Altor and classic variant. So that is your biotyping. Then you have antibiogram typing, which is practiced in hospital infection control. Then serotyping, like your Streptococcus Lancefield classification is based on serotyping. So phenotyping, you have bacteriophage, bacteriocin, biotyping, antibiogram typing, and serotyping. In genotyping, you have restriction, fragment, length, polymorphism, RFLP. Then you have ribotyping and pulse gel electrophoresis. Then you have sequencing also. You have sequencing technique to know the whole sequence. You can do whole sequencing, whole genome sequencing to look for any mutation in the bacteria. Thank you.
Best training, peaceful environment, PG medical entrance exam preparation were my expectations. I got them at Speed Medical Village in Kanchipuram. Today, I am a PG topper.
Speed Medical Village, a unique eco-friendly campus dedicated for PG preparation. Speed Medical Institute, a complete solution for medical education.